So just to review restriction enzymes and restriction sites. EcoR1 is a restriction enzyme that we get from E. coli, hence the eco, and that's restriction enzyme, hence the R, and it's the first one that we isolated from E. coli, so it's EcoR1. It's an enzyme that cuts DNA at this sequence, GAATTC, and it'll read the reverse on the opposite strand. And it chops the sugar phosphate backbones between A and G. So right here is where it makes a cut. DNA from another source with a gene of interest is cut by the same restriction enzyme and then added to the first. So here's our big DNA molecule that we're going to open up so we can put a new gene inside. We're going to cut it there, we're going to cut it there. And you can see it creates these sticky ends that I was talking about. And then we take a gene of interest from another DNA molecule that we cut out, and we just lay it right in there. And because it was cut with the same restriction enzyme, looking for the same restriction site, the same palindrome, it's going to hydrogen bond. Look, AATT, like that. Then DNA ligase comes in, seals the deal, and we get what we call recombinant DNA. And this is how gene splicing works. This is how we make transgenic organisms. Now, this red DNA here, the gene that we're inserting into the genome, for a long time, it was kind of clunky. We would cut out the gene that we're looking for, and it might have some extra DNA on other side because we couldn't be super precise with how we were cutting it. But recently, we have developed a new kind of molecular scissors, if you like, which is CRISPR. You may have heard of CRISPR in the news. CRISPR does not allow us to do... it's not what's allowing us to make transgenic organisms or do gene splicing. We could do that before. The thing that makes CRISPR impressive is that it is precise. Before, we were chopping things up with a butcher knife, and you can do pretty good things with a butcher knife. I mean, chefs swear by them. But CRISPR is more like a scalpel. We can get exactly the DNA that we're looking for, exactly the gene that we're looking for. And it uses a mechanism similar to something that we've seen before. Remember, we were looking at the microRNAs that could be created by the cell in order to attach to any mRNA sequence that we wanted to have a short lifespan, and it would basically guide a protein to break up that mRNA, right? It was just in an earlier video. CRISPR does something similar. So it has this Cas9 protein that it's attached to, and then we can make any kind of guide RNA that we are looking for. So this RNA is like a wanted poster. It's the description of the gene that we're looking for. And then the Cas9 protein can enter the nucleus, and it can run along the DNA searching for the precise sequence that we've programmed in, and it will make the cuts right where we want it to make it. So this is what I mean by it's precise. We can be very, very specific. We can switch out just a couple of nucleotides if we like. And then we can use that to splice in the new sequence. So that's CRISPR in a nutshell. It's gene editing technology, and the major thing that it brings is precision. In case you ever get the pub trivia question, what does CRISPR stand for? CRISPR. Clustered, regularly, interspaced, short, palindromic. Remember palindromes from just a little bit ago? It's talking about the sections of DNA it's looking for to make its cuts. Repeats. That will not be on an exam. It's just in case you get a pub trivia question and you need to remember it. So that's CRISPR. It's gene editing technology, but with the addition of this Cas9 protein using a guide RNA that we are able to program. We are able to synthesize any RNA that we want, and that allows us to be super precise. So, One way we can utilize this technology, we can take DNA from, say, a human cell, for example, insulin. We can take an insulin gene, and we can take the human insulin gene, cut it out, and then we can splice it into this little circle of DNA, which we call a plasmid. Most of the bacteria's DNA is in a single large chromosome inside the nucleoid region, but they can also pick up DNA from dead bacteria that are left behind, and usually those are in these tiny little circles, which we call plasmids. So what we can do is take one of those plasmids and use it as a vector, a vessel for carrying the thing we want. And the thing we want is the gene of interest, the insulin gene. So we splice that gene into this plasmid, and then we get the bacteria to take up that plasmid, and then as they reproduce, they start creating a whole bunch of insulin. And this is not hypothetical. This is the way that insulin is produced on the industrial scale for diabetic patients. So I said we were going to get the bacteria cells to take up a plasmid that we have genetically engineered in order to get them to change. This is actually something that we will do in lab. The Bacteria Transformation Lab, if you're in my Montgomery County Community College class, the Bacteria Transformation Lab is the last one we're going to do this semester. Here's how it works. We're going to take colonies of E. coli cells, and we are going to put them in a broth which helps them grow. So they're on this nutrient agar here. Then we're going to be transferring them into a tube which has calcium chloride in it. The calcium has a positive charge. It's going to attach itself to the DNA, and that's going to try to neutralize some of the charge of the DNA, which makes it easier for a molecule to get past the cell membrane. Because what we're trying to do essentially is get a chunk of DNA through the plasma membrane, and we spent a whole bunch of time talking about how hard it is to get big charged molecules through the plasma membrane. So this is what we're going to do. We're going to use calcium chloride to try to neutralize the charge, and then we're going to keep it on ice. We're going to really cool down those cells. You may remember this from the cell membrane material. Cells can change the ratio of saturated and unsaturated fats in their cell membrane. So once we freeze them, what they're going to try to do in order to keep a functioning membrane is to start putting lots and lots of unsaturated fats into that membrane. They're going to try to really increase the fluidity, make it more liquidy as a resistance to try to compensate for the cold. Then we trick them. We heat shock the bacteria. We put them into a warm water bath. Warm temperatures cause the membrane to become really fluid. And since it's already been converted to all these unsaturated fats, it's going to be super fluid. So that's going to be our best chance to get the DNA through that cell membrane. So the calcium chloride, the chilling, and the heat shocking, it's all just to try to get the DNA plasmid into this cell. Now, on that plasmid, there's going to be a couple of genes. So we're going to make some genetically engineered organisms. One gene that's on there is for ampicillin resistance, an antibiotic that's going to resist. We're going to plate it on this plate here that has the antibiotic built into it. Now, why? Because not every single cell inside this culture is going to take up the plasmid, and we only are interested in the ones that did. So all the cells that took up the plasmid will have this ampicillin resistance gene, so they should be the only ones that are growing on this plate. The other gene that's going to be in there is called GFP. It's a green fluorescent protein, and that means that under a black light, it will kind of glow for us. It'll glow a little bit green. So any of the cells that manage to grow on this agar, since the plasmid is just one chunk of DNA with a couple of genes on it, anything that is able to grow on this ampicillin agar has the plasmid, and it should also glow in the dark. So we're going to then put them in the incubator grow up our cells, and then see if we can get the bacteria to glow. This whole thing is to make some glow-in-the-dark bacteria, but really it's a demonstration of how we can transfer genes from one species into another. The glow-in-the-dark gene is from jellyfish, so we are creating some transgenic organisms here. Okay. And this will be kind of what the results will look like at the end, but we'll go over that more in lab.